talking about the cell in chapter 4, how it works, right? Chapter 3 was all about the pieces. And this is about how it works.
So our test is over 2 chapters?
It is. Yeah, it's been on the sheet for 2 weeks. You're getting pieces. Don't worry. That's the way it rules. I'm sorry. There's no other way to give it to you. All right? So here is this first section. And what I want to make sure you understand, if you look at your study guide, the places where the WAU piece focuses on: organelles, types of membrane transport, meiosis, mitosis. And then here's the drawing, the line between chapter 3 and 4. They skip all of this part. There's none of this part that we're talking about today that's on their test. And hang on, and I'll get to you. And then they ask you about protein synthesis, which is the last piece of chapter 4. So I'm telling you that I am not looking for superb amounts of details here. Okay, I'll be very clear as far as what I'm looking for here. And this is a lot of materials, 2 different chapters worth. Okay? And it's stuff that you need to be exposed to. You need to see this stuff. You need to have some grasp and understanding of it. But I'm not looking for the intricate details in these. Does that make sense? And I'll give you some more details as we go through. Yes? Just right. We're talking about from metabolic reactions down through DNA. And again, it's stuff you should have seen before. There could be maybe, well, I'm not going to give you numbers, but there'd be just a couple of questions there, more focused other places. Your question. Characteristics of cancer, there's a fantastic piece. We did not spend much time talking about it, no. At the very end of chapter 3. I'm sorry, I'm losing my spot here. Where are you at? Thank you. I just can't find it this morning. There you go, on page 78. There are 5 pieces. Hyperplasia, which means it's just uncontrolled cell division. It's just dividing like crazy. Dedifferentiation is on page 79 if you want to look at it. Dedifferentiation means instead of it being differentiated, remember our stem cells, right, become progenitor cells, become actual cells. Cancer cells dedifferentiate. They're like progenitors or stem cells. They're not specialized, right? The third one is invasive. It's going places it's not supposed to be, right? Here are some skin cells that are, in some reason, going crazy and going down into the connective tissue and into the bones and muscles. You with me? Angiogenesis means it gets its own blood supply. It actually starts growing its own blood supplies, because it needs blood, right? These are cells, they need to work. And then last but not least is metastasis. It spreads other places. So again, I just simply want you to be comfortable with those 5 pieces. Sir?
What's the difference between cancer and a benign tumor?
So a benign tumor will have some of these characteristics, but not necessarily all of them. Especially the aggressiveness of the metastases and invasive. So it can be just a set of cells that for some reason have started to grow extra, but they aren't necessarily looking to invade other places or spread other places. They're just kind of localized in one spot. It's a good question. There's a whole lot more detail than that. I'm giving you the super simplified version, but yeah, that's the key. Sir? Right, in other words, it's not aggressively seeking to spread other places. That's like the simplest definition between benign and cancerous. Okay, thank you. I appreciate you pointing that out, that we didn't talk about that piece. So when we're talking about metabolism and how things work, there's really 2 equations or 2 sets of reactions that happen. The good news is you already know them. You just know them as different names. You know them as synthesis and decomposition. That's what we learned in chemistry, right? When we're talking about in an A&P setting, in the body setting, instead we use anabolic and catabolic. Anabolic means putting together. Catabolic means to break apart. Okay? And that's really the basic pieces of what I want you to understand. There is a little bit more detail there that your textbook provides, that anabolism is actually what we call dehydration synthesis. So if you think of it like a category, you and I would have called that synthesis, right? Back in chemistry, two becoming one, the married couple, right? A better name for it is anabolism. A specific name, like of the actual reaction that's occurring in your body, is what we call dehydration synthesis, where we're adding two things together by taking out a water molecule. And this is a picture that's showing you that. Here are the two monosaccharides, right? Our simple sugars, our simple carbohydrates, coming together to make a disaccharide. And then look what pops out, a water. So dehydration synthesis, right? Does that make sense? Oh, no, this is synthesis. This is coming together. This is anabolism. Catabolic is breaking apart. Anabolic is coming together. Yes. And again, they sound very similar. Just the C and the T in the end. There's just little differences between them. Okay? And again, the point of that catabolic reaction is to release energy. You're breaking things down to get the pieces from them. Things that are formed via dehydration synthesis, polysaccharides, so again, these multiple sugars, lipids, proteins. You don't have to worry about the different peptide bonds, a dipeptide or polypeptide. I'm not worried about those. Just being comfortable. Anabolism, two things coming together. Dehydration synthesis is the specific kind. Catabolism, again, breaking apart. The specific kind of reaction is hydrolysis. If you look at the words, hydro, which means what? Water, right? All you all Marylanders say it, water. And lysis. What does lysis mean? Like lysosomes? Like break down, like breaking apart, like Lysol, killing all the bacteria. So this is hydrolysis. So we're using a water molecule to split a bigger something into two smaller somethings. Does that make sense? They don't show you on the screen here a picture of that. There is a picture that shows you a bit of that there. That is on page 89 on the very top. I'm not sure why they didn't include that one here, but it is there for you as well. And again, they show you a couple examples. On page 89, there's another example of that dehydration synthesis between making a dipeptide model on the bottom there. Again, key pieces. We have synthesis, we have decomposition, right, in our bodies. We're breaking things down, we're adding them together. Adding together things we call what? Sorry? Anabolism. Right? And then breaking things down we call catabolism. The actual reaction that anabolizes, that put things together, what do we call it? Dehydration synthesis. Absolutely. Right, which is the same as we started with, right? Synthesis, things coming together. Okay? And then the thing that breaks it apart is hydrolysis, hydrolysis. Using the water to break it apart.
So catabolism is just coming together, and dehydration synthesis is how it does that?
Yes, it's like the specific name for it.
Okay.
Yep.
So there's hydrolysis and lysol?
I'm saying it was basically the same name. It's the same root of those words. So, lysis meaning break apart, kill. Exactly. So in our body, the reactions that we do, these anabolism and catabolism, are helped out by these things called enzymes. In our chemistry terms, what is an enzyme? It's a protein, certainly. It's a catalyst. You're going to say that, I know. A catalyst. And remember, a catalyst does what? Give me a raised hand. What is its point? Sir? It speeds up the chemical reactions. But not necessarily just speeding up. There's a more technical way that we talked about what a catalyst does. Yes, ma'am. It lowers the activation energy. Remember we gave the example of the big pile of woods that sit out here on 198 at these landscape folks? I mean, that's burnable stuff. So why isn't the fire department all worried about that? Well, it has a very high activation energy, right? Now if I went and dumped a whole bunch of gasoline on top of that fire, or on top of that wood, okay, now they would be a little more nervous, right? Because the activation energy is so low, one little spark set the whole thing off. So enzymes are things that allow your body's reactions to occur at a much easier rate. It takes less energy. And an enzyme is simply a biological catalyst. Instead of just being something in chemistry that we mix together, a different other compound, this actually is a protein that fulfills this role. And here's how it does it. And again, I think that this picture that they have for you is such a solid one. Where are we at in your textbook here? Thank you. On page 90. What you have are these two substrate molecules. Think of them as the guy and the girl who like each other, but like don't want to talk to each other. You know, they don't roll in the same crowd, so they're kind of, these people know that she likes him, and these people know that he likes her, but they don't actually ever get together, right? So the enzyme is that friend who like kind of is involved with both groups, and they're like, hey, you know that she likes you, right? And they say to her, well, you know, he likes you. You guys should get together, you know? And so these two substrate molecules come together, and can you see that there is a specific pattern on top of this enzyme, what they call the active site? Because each enzyme is only for a certain reaction. So these two substrates come together. The enzyme allows them to chemically bond, react together to form this whole new something, and then it lets them go. You can almost think of the enzyme like as the pastor at a wedding ceremony, right? Two different, a groom and a bride coming together. They are becoming one flesh. The pastor blesses them. Let what God has joined, let no man tear asunder, right? And then he sends them off on their way, and he gets ready for the next couple to come. Maybe there's like a Vegas chapel where they just roll people through there all the time, right? But are you with me? The actual enzyme is not changed in this reaction. The enzyme does not play a part in that reaction. It would be a little weird if there were three of them getting married together. Right? It's just simply the two substrates coming together. Does that make sense? Okay. And just about every reaction in our body is governed by these enzymes. And sometimes even this whole series of enzymes, this metabolic pathway, where I'm going to take this. Think of it like you're building a car. That these guys here are going to be the enzymes that allows all of the wheels to come together on the axes of the car, right? And then they pass it along here, and these ladies put the body on it. And then they pass it over here, and then these guys put the seats in the car. And then they pass it over here, and these guys put all the interior pieces, all the monitors, and the touch screens, and the radio, and right? It's basically just this assembly line that rolls through, what they call a metabolic pathway. And are you comfortable that if these guys on the end here mess up the tires, then the whole thing doesn't work, right? These guys are controlling that process that we're trying to undergo here. Does that make sense? Sure. I mean, they would frankly keep making more tires because we're going to keep this chain going. Yeah, so that's how a factory works, right? These guys just spend their whole day making tires and putting them on axles and shipping them, pushing them down the line. Does that make sense? Yes, ma'am. Oh, sure. Anywhere along there could be a problem that would undergo it. But we're basically saying that this first guy up here is who controls everything. He's what they call in your textbook the rate-limiting enzyme. You don't have to know that term, but that's what they call that. They determine how fast. If these guys only make one axle a day, you're only going to make one car a day. If they make 1,000, well, we could maybe make 1,000 cars. Does that make sense? So that first enzyme governs how fast we make cars. Are you with me so far? Not moving too fast? I'm purposely trying to talk a little bit slower, a little more common. I understand. It's tough. There's so much stuff to get through with you guys. I appreciated there's about 2/3 of you who actually gave us some feedback on those surveys. If you didn't, well, you lost a couple of points, frankly. But I would still like you to put it up there for me. Because I do want your feedback. I want to hear from you. Schoology, my friend. Yeah, Schoology. All right, last but not least, today, we're going to talk about how your body makes energy. And, of course, energy is just how we do work, the capacity to do work. That's a physics definition of what energy is. And you've got lots of different kinds of energy, right? Heat and light and sound and mechanical energy, electrical energy, chemical energy. All of these are the same. That's what's one of the crazy things of Mr. Einstein's equation, right? E equals mc squared. He says energy and mass are the same things. Wait, what? You can take mass, actually like this. I don't know what this is. What is it? A pencil case. Good. I felt like it had water in it or something. It's cool. Very cool. And you can turn that into energy. And realize that that's what a bomb does. That's what a nuclear bomb does. It takes this energy, this mass, these tiny little bits of radiation and radioactive material, and turns it into energy. Like this little bit, well, I thought there was water, so I'm going to steal some water. Is there water in here? This water right here, this is about maybe, I don't know, maybe a cup and a half, 2 cups of water. You could power a whole city with this water.
Are you serious?
I'm not kidding. Yeah. If you could take this mass and turn it directly into energy. Okay? Which we are not even close to being able to do that. But that's how much energy is contained there. Yeah. All right, so we moved on. We got a little sidetracked there. And the process that we're doing, we're using these chemical processes to make energy. What does the cell use for energy? Do you remember? ATP. Adenosine triphosphate. And the whole goal when you eat stuff is your body has to take that stuff you ate and turn it into ATP. As a matter of fact, what they're doing is they're taking ADP, adenosine diphosphate, and they're adding another P on the end of it to turn it back into ATP. They're like the little rechargeable batteries of your cells. They go from ATP to ADP to ATP to ADP over and over and over and over again. Okay? So this is the process. Will you take a look? Where are you at? Where am I at now in this lovely textbook? This is on page 92. And your chart will look just slightly different than mine because I have the older textbooks version. Yours is good. Yours is actually better, how they did a little bit of changes to it to make it a little bit clearer. Those of you who are moving on into medicine, you will learn this in much more detail than I'm showing it to you now. As a matter of fact, one of the first big tests you have in Intro to Biology is on this, with, again, significantly more detail than I'm going through here with you. Even though we're going to spend some time talking about it. But there's a lot of stuff that happens here. Okay? And again, the whole point of this is called cellular respiration. The whole point of it is to make what? ATPs. We're trying to get energy from what we've eaten. Are we cool? Sorry? Through a lot of work. It's actually quite interesting that each of these little processes we'll talk about, somebody studied and figured that out. Like their life work, yeah. 20-some years of work to find out that the glucose breaks into 2 pyruvic acids, and we call that glucolysis. Yeah. Somebody studied that for years and years. It's quite impressive. We certainly stand on the shoulders of giants. That's the old saying. All right, so there's 3 pieces to this cellular respiration. The first is called glycolysis. So let's look at the word. Glyco meaning what? Glucose. Glyco is sugar, right? Glycolysis. What does lysis mean? Breaking. So we're taking sugar and we're breaking it. Again, scientists are not very creative. Okay, the words tell you what's happening. Glycolysis. Either that or we just number things. And we give them these numbers that look all impressive. Yeah, that's how we roll. Yes, ma'am. Yeah, taking a sugar. So in this case, we're taking glucose, which again is a very simple carbohydrate, right? It's a carbohydrate that we've taken our other carbohydrates, our complex carbohydrates that we've eaten, and we're breaking them down to glucose. That's a whole other process. That's what your digestive system does, right? Breaking down those pieces and making them into these simple sugars so your body can use it. We notice that glucose has how many of these black dots with it? It's got 6. There's actually, in a glucose molecule, there are 6 carbons. And one of the keys for this process is follow the carbons. Okay? Because we start off with 6. And you're going to see as we move through that those 6 become 2, become 4, become 6, this big cycle of how they work. All right? They do. So let's follow the carbons. So glycolysis means that 6 carbon sugar is broken in 2 to make 2 pyruvic acids. It's just the name of this acid. Yep. Breaks it into 2 pyruvic acids. When it does that, it creates how many ATPs? Friends, how many ATPs does it make? 2. Again, these are pieces that, these are all the things that I'm pushing you to be comfortable with, so stay with me. It breaks into 2, it gives off, the energy is created to make 2 ATPs, and then it releases something else that goes off to the left there, following the blue arrows in your textbook. High energy electrons. Okay? And that is part of the third piece down here, the electron transport chain. So we'll come back to that, but I want to make sure you know that there are high energy electrons that are released during that glycolysis. So 3 things, right? Glycolysis breaks the glucose and makes it into 2 pyruvic acids. It creates how many ATPs? 2. And it gives off high energy electrons. Sir? Yes, we'll get to that. We'll get to that. Yes, we'll get to that, definitely. So that was step 1. That happens, it says, in the cytosol, which is basically meaning outside, like inside the cell, but not in any particular place. The cytosol is the actual name of the fluid that makes up the cytoplasm. Right? The cytoplasm is the inside of the cell. Remember, right? Cell membrane on the outside, nucleus on the inside. So don't get confused. Cytosol just means cytoplasm. It happens outside of any particular organelle is what they want to make sure you understand. We're cool so far? Glycolysis. Glycolysis, then, one of those pyruvic acids is taken into the what? This is the Krebs cycle. Sure, that's the name for this whole kind of piece, but where does it happen? What organelle? In the mitochondria, because the mitochondria is the what? Well, you know this, the powerhouse of the cell. It was ingrained, I think Mrs. Serrano woke you up at night, 3 o'clock in the morning. What's the powerhouse of the cell? Mitochondria. Right? It's good. But know what that means. This is the why. You want to know why it's the powerhouse of the cell? This is it. This is what it's doing. So it takes one of those pyruvic acids at a time. They both come in, but this cycle is just with one at a time. That's why your diagram only shows the 3 carbon there. It just says 2 of them. Mine actually has 2 up there. They both go in, but just one at a time. Okay? When it comes inside the mitochondria, this is what we call the citric acid cycle. This whole piece right here. That pyruvic acid loses 1 carbon, right? So it was 6, right? It's split in half to make 2 3s. One of those 3s, it loses 1 carbon. That carbon goes off and makes a carbon dioxide. And we're left with a 2 carbon. It's called acetyl coenzyme A. And we abbreviate it acetyl-CoA. It's good. You sound smart when you say these names, by the way. Pyruvic acid, acetyl-CoA, they're like, oh, wow, you're impressive. They're just names. So we lost one of our carbons. That's our carbon dioxide. Remember how we talked about when you breathe out, you're breathing out more carbon dioxide than you took in. And that carbon dioxide is from the metabolism of our cells, right? It's the waste products. This is what we're talking about. This carbon dioxide right here that was created during this citric acid cycle. That acetyl-CoA now meets up. Again, it's a 2-carbon now, right? Remember, it was 6 as a glucose. It broke into a 3-carbon, which is pyruvic acid. That 3-carbon came into the mitochondria, lost 1 carbon to become acetyl-CoA. And now, inside the mitochondria already is this 4-carbon acid called oxaloacetic acid. It was already there from the previous, because it's this huge cycle that keeps going over and over again. I would have to assume that that was what God had to have done, was to start the process. Yeah, but since you were born, even before you were born, these processes have been undergoing. Even the two ovum, the ovum and the sperm had these processes in them, and it just kept rolling. So the acetyl-CoA 2-carbon meets a 4-carbon oxaloacetic acid, and they bond. And they make a 6-carbon citric acid. Whew. So let's review again. It went from 6-carbon glucoses, right? They were together. It broke apart to make what? 2 what? Pyruvic acid. Like Pyrex, like the stuff your mom measures stuff in. Right? Pyruvic acid. One of those comes in, it loses a carbon, it becomes acetyl-CoA. Good. That acetyl-CoA bonds in this citric acid cycle with a 4-carbon oxaloacetic acid to make a citric acid. Okay? By the way, I forgot to mention, when that 1 carbon comes off of the pyruvic acid, it also releases high energy electrons. Forgot to mention that piece. Give me just one more second. That citric acid then gives off 2 more carbon dioxides to get back to that oxaloacetic acid. The oxaloacetic acid then grabs another acetyl-CoA that's come in. It makes citric acid. 2 carbons come off. It forms oxaloacetic. And we just keep rolling this process over and over and over again. There's actually about 20 different steps in that citric acid cycle that you'll learn in Intro to Biology. We're not going to go there today. Okay? But there is way more detail than I'm sharing with you here. Your question?
So when you have the pyruvic acids, the 2 of them,
yep.
Only one of those goes in?
One at a time. Oh, okay. So this is basically showing you one coming in and dealing with it, but the next one then follows right behind it. Yep. In that citric acid cycle, how many ATPs are created? 2. 2 in that turning of taking the acetyl-CoA and oxaloacetic acid and making citric acid. That's making 2. So we had 2 that were made where? The glucolysis, right? We have 2 that are created in the citric acid cycle. And then the last piece of this whole thing is the electron transport chain. Whew. And I know this is a lot of stuff. I know you're bored. I got you. Stay with me. You see there are 3 places in this cycle where we release these high energy electrons, right? At glycolysis, when we cut off that first carbon dioxide from the pyruvic acid, and then when we're doing the citric acid cycle. Those are the 3 times we get these high energy electrons. These electrons form this electron transport chain, and they bring all these electrons together. And it's quite a process. Again, they're super simplifying it here for you. And what those high energy electrons have most of the energy that were contained in the bonds that we've broken apart. All these different places. Up in glucolysis, when we broke off that carbon dioxide from the pyruvic acid, when we break apart the 2 carbon dioxides from the citric acid, these high energy electrons are holding all that energy. And they bring it down, and through a series of enzymes, the reactions, they take those high energy electrons and store them as ATP. Here's the little piece that shows you how that happens. Again, this is one page back, I think, in your textbook. Yes, on page 91. That you have an ADP that comes into this citric acid cycle. And the high energy electrons are used to bond another P. You see how there's 3 P's here, but only 2 P's there? It's this bond right there that they're trying to form. To take these 2 P's and add that third one onto it. Again, think of it like your batteries that you're recharging on your phone every night, right? Or sometimes throughout the day over and over again. That energy is being stored in that bond so that then that ATP can go out to a cell. Let's say I'm the ATP coming to Eric, the cell. Eric would break off that 1 P. He breaks it off, and that releases the energy that allows the cell to do whatever he wants, right? This ball of fantastic energy that is amazing. Yes. That ADP then gets sent back to the ribosome, and we make it again. How efficient is that? Well, take a look. How many ATPs do we create? 32 to 34. So if we do the math, right? We did 2 way up in glycolysis. We did 2 ATPs in the citric acid cycle. We did 32 to 34 in the electron transport chain. So I add all those up, and I'm at 36 to 38 ATPs from 1 molecule of glucose. That's extremely efficient.
Wait, you said 2 in glycolysis.
Right? 2 here, 2 there, and 32 to 34 there.
So all of it pretty much comes from the high energy electrons?
That's where the vast majority comes from, yes. You're correct. Whew. Are you with me, friends? It's a lot. I'm not kidding, it's a lot. We'll spend some more time with this. Okay? Yes, ma'am.
That guy that does a lot of the videos that you post.
Right. He does. I'll post something up there for you. Yep, certainly. Certainly. Okay. So, when I see you again, because we do have another class period today, we're not going to talk about this. We'll let this marinate. I would encourage you, though, tonight, go back through that. Shh. You spent time with it over the weekend, or at least you were supposed to have. Go through again tonight. And again, roll through those steps. What's important is, I do want you to be comfortable with the names. And I know they sound all fancy and stuff, but be comfortable with the names and what's being created, right? That we're releasing how many ATPs. There's electron transport chains that's happening. There are carbon dioxides that are breaking off. Are you with me? And again, what's the whole point of this? Is to make ATP. That's all what it boils down to.
When are we going to see each other again?
Today, this afternoon. Shh. We're not done, guys, so don't start packing yet. We're here together in period 3 at 1:10. What we're going to do is a case study. We learned all about how cells transport things and how it works, but that's very abstract. I want you to see it in real life, like on a real person. What would it look like? How do those transport mechanisms actually play a role? We're talking about Botox, actually. You know, like you're all wrinkly and you want Botox. So that's what the case study is on, a lady who is dealing with Botox. We'll talk about that this afternoon. Okay? You are good to go. Adios. Again, remember to be quiet, please.